This presentation is the work of the Dr. Hayley Everskin, that, as I told you, she works now for UMedSat. She was before in PML, and yeah, and her PhD in South Africa. So there are some other collaborators that are mentioned in this first slide, and I'm going to just communicate what she did and also to talk a bit about what are these harmful algal blooms. So the agenda is this. I go rapidly through it because we need to catch up. So the first thing we need to think about is what is a bloom in itself, because this term is not very well defined, and many people defining it in a different way. When do we have an algal bloom? What is the right quantity of algae we need to have to? What is the right quantification to consider there is a bloom? So I asked ChatGPT, because it's a new thing, and ChatGPT said an algal bloom or algae bloom is a rapid increase or aggregation in the algae population of the freshwater or marine water systems, and is often identified by water discoloration of their pigments. So we know that these pigments are mainly the green or the red reflectances, and that's why we identify them. But it said nothing about quantities of algae that we need to have there, or the timing on when these blooms occur, the season of the year. So there are other considerations we need to take into account if we want to define properly what is a bloom. And then we have to add this harmful adjective to the bloom. So a bloom is a concentration of algae that cause harm. So we need to think on the impacts of these blooms, what is the way they can harm, and try to quantify using metrics applied to the data we have. So typically, when we think about an harmful algal bloom, I'm going to say HAP from here on just to shorten it. We think in a mass quantity, a high biomass, mass quantity of algae in the water. But this is not always the case, as we will see in a bit. So some of the impacts of these hubs, there are here like 8 different ways that a bloom can harm. It's like through starvation, through mechanical or physical damage to the ecosystems, to anoxia, absorbing all the oxygen, through the toxicity of the ammonia, through phycotoxins that are in the algae, and through growth inhibition, what is called allelopathic damage, or ambush predation. So to generalize all these 8 different ways in 3 big groups, we can say that the hubs can cause anoxic events. And usually these anoxic events, this lack of oxygen, is due to high biomass, or high dense algal blooms, to toxic events. In this case, we don't really need to have a high biomass in our algae. We just need to have toxins inside this type of algae. So we need to identify with which type of species carry these toxics or act toxic. And the ecological harmful events, when there is some crossover, so there is a damage in the ecosystem, and it's maybe not direct. In some cases, we don't even know we are being intoxicated by algae, because it's fish that has been intoxicated that we eat, and then we have a bad stomach for a few days, and this is also an intoxication by algae. Algae, some of the toxins can also be aerolized, so if there are the proper winds and go through the coast, they can also arrive to our respiratory system. So to be aware of these events and to prevent them and to palliate the damage they cause is really very important. What are the monitoring requirements for hubs? We need, of course, the dream of all environmental scientists to have a lot of data with good spatial and good temporal resolution. These blooms usually occur near the coast, so we need this high resolution. It's really beneficial, and also usually some of them have a regular or irregular frequency that we also need to monitorize. That means we need both good temporal resolution, good spatial resolution. We also need to understand the characteristics of these blooms. So some of these hubs, we said, are linked to high biomass. Some others are not, are more related to the species that compound or that form these blooms. So we need information about individual species, about the function of these different species, the size, and of course we know all these characteristics, or among all these characteristics, the color is one important one because we have this capacity of distinguishing colors in the water. Also, we need to understand why why a hub is happening. What is the driver of this hub? If it's a natural cause or if it's an anthropogenic cause, we need to be aware of these things. So I will start just mentioning these non-natural causes of hubs. So we have an introduction of nutrients into the marine environment from activities that we do, anthropogenic activities. For instance, with the waters that arrive to the coast from our sewage systems, or because of the increased growing land dedicated to agriculture that carries a lot of fertilizers into the coastal waters too. And I wanted to read here in this this slide. It says there's a link between the trends in fertilizer use and the number of red tides for Chinese coastal waters. And annual fertilizer uses has climbed 156 million tons in 40 years, and it continues increasing. And when we have a strong natural buffer zone, when we have a forest, when we have wetlands, and a diminished runoff of nutrients, there is a clear decrease in the appearance of these hubs worldwide. And you have here a map with some of the areas where more of these types of hubs have been detected. But sometimes these hubs have a natural cause, mainly due to the introduction of nutrients from deep waters by a physical oceanographic process called upwelling that you all know. So paradigmatic cases are Peru, Eduardo probably knows, or the Benguela currents or the Benguela area to put some examples. So what Haley did for her thesis was to try, hope I explain it properly, so to try to understand what are the different variables in the ecosystems and to compare that with the locations where the blooms appear and the species that are identified by correlating all of these variables. So this is what we have here. So that's a way of trying to understand which types of hubs are we going to have depending on which currents, which winds, which locations. So it's not an easy task as we see, but it's an important thing that we need to understand. So from the remote sensing point of view, so far we have been talking about hubs, but we are not really making reference only to remote sensing data. We can also use in situ data to detect them. But concerning the challenges for remote sensing, the main problem that we have is that there's something in my eye, that we don't really have a good balance between the spatial, temporal, and spectral resolution of the sensors to be able to understand what happens in the water. So the sensor sensitivity is really very important. For instance, when we have a medium to high resolution sensor like Sentinel-2 or the Landsat series, we don't have a very good signal to noise ratio, which yeah, we need to compensate somehow. And then all these events usually happen in coastal areas, and this complicates a lot of the things. So we have complex waters in complex areas. So if we want to use remote sensing, we need to take care of the atmospheric correction, that my atmospheric correction is really doing a good job in these areas, to take into account the adjacency effects that we have seen, the sun glint effect that also has been mentioned. And then we try the blue-green ratio algorithm, and it's not working properly because my bloom is out of range for this type of ratios. So in general, for remote sensing, there are many complications. Well, in this image here, this is from Haley's thesis. This is a Mary's image from 2005. And exactly the part where the bloom is happening is masked, because the algorithm they were using at that time was not really prepared for this high, big quantity of biomass in this area. Right? So specifically for hubs, you have seen during the course, and you know about those things more than me at this point, is that if we go to the lab and we have different types of species of phytoplankton, we can derive a signal in the remote sensing that is quite distinct. So we are able to differentiate in between or among different types of species. But we also know that when we want to apply an inversion algorithm to go from remote sensing to species, there is no direct relation. We have in statistics, it's called, I think, an ill-posed problem. We have the same signal for special different or many different combinations of things that are in the water. So this is one of the main problems, and it aggravates somehow because usually with remote sensing, we don't have the hyperspectral capacities of the labs, even though we know now that there is one operational hyperspectral satellite that is flying that is space, and we hope that we can use that for this type of problems. But so far we had sensors with 13, 20 bands and with really broad wavelength ranges, and everything is really confused. So to identify exactly the peak that makes a distinction between two different types of phytoplankton species is really complicated. So the best approach usually is to combine remote sensing, different spatial and spectral resolutions with in situ data, and try to do a hybrid model and work with that. And there is no one size fits all solution. So it's really very localized for the type of bloom I want to study on the location I want to do it. So the hubs research is very nice field of research for ocean color remote sensing scientists and ocean color scientists in general, because it's a sometimes natural, sometimes anthropogenic circumstances, but we can mix, let's just say, the all the knowledge of the ocean color to try to deep further in this problem. And it's also a good opportunity to use synergistic techniques of remote sensing. So because we know we are going to need more than the ocean color, or more than the chlorophyll, or more than the absorption of my species, I'm going to need winds, I'm going to need temperatures of my water, I'm going to need currents. So it's a nice way of doing holistic, close to holistic approach, of research in environmental marine sciences. Um, the benefits of starting this type of researcher approach, of course, for me, would be more important the part of the health, how important it is for us to have clear and healthy waters where that we can drink, or where we can go there to the beach and bath without any problem. But it's also important for the industry, for the tourism industry, and also for aquaculture. So we are more and more people in this planet, and we are going to rely more and more on aquaculture. And this is a reality, it's already happening. And one of these HABs events can cause a lot of damage to this industry. And there was a case like a few years ago in Chile with huge losses, until 80 million of dollars lost from a single event of hubs. So some approaches to face the problems of the hubs using remote sensing or hybrid methodologies. For instance, when we want to study high biomass hubs, we need to move from the blue-green ratio algorithm to other parts, to use other bands, other parts of the spectrum. And what we have here is that in this work, what they did was to, by increasing the chlorophyll A content on the water, we see that the peaks of the reflectance moves to longer wavelengths. So instead of using only the blue and the green bands, we can also use the red H bands, or the infrared bands. So some of the algorithms that are used in this case are summarized in this publication. For instance, using the fluorescence line height can help to improve the detection of these types of high biomass blooms, or the maximum chlorophyll index, or the maximum peak height. There are different ways of approaching this type of problem. And in this image here, we see that area that we couldn't capture with a blue-green ratio before in the image of Mary's, is now showing that there is a bloom here in the coast of the Benguela. For species detection, we need to know very well which models we are using to which biooptical models we are using to derive our data for um, training algorithms, for instance. So we know we need to understand what I said before, how distinct are the properties you want to look at. So how much is the biomass that you need to be able to see the particular pigment that is causing this peak or this change in the cell size that is going to be the clue to distinguish between different species, or how big is the change in the cell size to produce this change of detection in the signal. So we need to understand very well what we what we have and what we can see with the data we have. Again, I repeat, with hyperspectral, very detailed data, we will be able to see a lot of things. When we go to Sentinel-3, Sentinel-2, Landsat, and the sensors we are using right now, the number of bands is less. The capability of distinguishing between species is reduced dramatically. And also we need to understand the uncertainties we have in our in situ, in our remote sensing sensors, and the errors that are produced. When we want to study the environmental dynamics, as I already said too, it's good to have to take into account all those variables that can be affecting the cause or the appearance of that bloom. And for instance, in this case, this is Haley's work too. They knew that in the Benguela current, there are upwellings every year, and it's seasonal. But not every year there is a hub. So why in some years there is a hub and in some others there is not a hub? So you need to understand what are the physical variables that can be helping to these hubs to be developed. And they did an analysis on the currents. So in this case, the coastal currents north to south, the Jaya here, and also on the winds that were affecting when the blooms appear. And they managed to, well, I wouldn't say forecast, but when these blooms start to appear, when these wiggles here start to show up in the north, so that are related to certain types of winds and strong currents. So, or strong, I mean, not strong, but the potential of these currents. So they know that when they start appearing in the north, they will move with a velocity, certain velocity to the south. So they can, if here, for instance, we have aquaculture fields, we can tell the people harvest your your fields before 2 weeks because a hub is coming and it will destroy your product. So do it before. This is a way of somehow predict or alert what is going to happen. So there are some operational already in our community and in certain countries, some operational services for hubs. NOAA has one of these hub services, and they use Sentinel-3 data, and they also have this like respiratory forecast, so they know the species that can cause this, that can be aeroolized and then can cause this type of problems in the health of the people that lives near the coast, and they can send an alert. Or one of the colleagues of Haley, Matthews, also developed this alert for cyano blooms in lakes. And this is also an active warning for cyano blooms. Or also in the north of Europe, Brockman Consult and other partners have developed the cyano alert, and they are still working on this type of products. And also PML in UK has a hub bulletin with prediction of blooms in the coast, and they also study the currents. So, and this information is really very valuable, especially for the insurance companies to know the risk of something happening. To sum up, there is a great natural laboratory for applied optics and remote sensing research in hubs. But we need to understand very well what we consider is a hub, quantify it, what is it depends, as I said, on the location and on the characteristics of each place. We also need to understand what is the sensitivity of our signal with the sensor we are using, because there are limitations and we have to be aware of these limitations. and it's a good opportunity to try combine approaches like using in situ with remote sensing or using some modeling to estimate the risks. There are some reference here, including the work by Haley and other important references. And there are all other links here. For instance, with this work. So Haley shared some material from her Google Drive. Maybe it's not working. No, because I need to be sign in, right? So there are an extended, this lesson has been extended. There is a PDF with a lot of information and the images that she used and an activity you can do by yourself. We are not going to do it now. UMETSAT also has generated case studies, and these case studies have Jupyter notebooks to go through them. And of course, UMETSAT is open to cooperation. If you have a nice case study you want to share with, it will be reviewed and published. And this is the newest one, the oxygenization impacts on marine life in the Benguela area. But there are other user cases in Europe and Africa, also from the UMESAT user portal. Of course, it's very important to mention the IOCCG report on hubs from 2021. You know that IOCCG have several very important reports, very pedagogical and on the state of the art of several topics on ocean color. And we also preparing 2 new application user guides that will be uploaded to the UMETSAT user guides user portal too, on hubs in general and cyano blooms in particular in probably early next year.